Hello, everyone. I hope you guys are doing well. My name is Anish Ghosh, and I am currently a sophomore at Providence Day School, and that is in Charlotte, North Carolina. Some interests or hobbies that I have, extracurriculars, are I play violin quite a lot, and I also enjoy doing robotics. So this is my introduction. The topic I have decided to research about is regarding nanoparticles and the use of nanoparticles in targeted delivery of drugs for lymphoma using copper-free click chemistry. And copper-free click chemistry involves a chemical click between an azide and an alkyne, which is shown here. And basically, what happens is the in this case the alkyne is a chemical called DBCO, which stands for dibenzocyclooctane. And as you can see in the image, the DBCO attaches to the azide here, and it basically forms a triazole ring, and the reaction is called a SPAC reaction, which stands for stain-promoted azide alkyne cycloaddition. And basically, what I wanted to research about was whether this copper-free click chemistry can be used for nanoparticle-based drug delivery in lymphoma. Basically, the need for finding this kind of method is to minimize toxicity, because current treatment methods such as like chemotherapy harm healthy cells as well when patients are treated with chemotherapy. It, along with harming and killing the lymphoma cells or the cancer cells, it also harms healthy cells, which is basically the whole point of the nanoparticle targeted delivery of drugs, is to minimize that toxicity. So the literature review that I did for two papers, both of these papers are about the use of nanoparticles for copper-free click chemistry for the treatment of non-Hodgkin's lymphoma. And the first paper discusses the use of nanoradioimmunotherapy. And basically, what it talks about is how currently there's pre-targeted radioimmunotherapy is used, or it's being tested in certain preclinical models. However, the issue with this with pre-targeted radioimmunotherapy, which an acronym for it is PRIT, the issue with it is that it results in immune responses as well as interference with other molecules. So basically harming other cells which are not meant to be harmed. And so this is where the nanoradioimmunotherapy comes in. And this kind of method, with this method, the paper saw that the radioactive material was successfully sent to basically it was a CD20 antigen, which was used for in the lymphoma cells, and it was able to successfully send the radioactive material, and it was also able to limit the damage just to the lymphoma cells, which is one of the major points of it. So by being able to do that, it was successfully able to not prevent harm for healthy cells. So, and then the next paper is it's mostly about the same concepts being used, except instead of radio immunotherapy, it talks about small molecule inhibitors. And these inhibitors, they face challenges such as poor cancer targeting specificity and also significant on-target and off-tumor side effects. So these small molecule inhibitors have led to effects such as hepatitis, colitis, and pneumonitis. So this paper, the second one, it tested using nanoparticles on mice, and with the use of nanoparticle formulations, they finally saw that the mice did not have any significant damage in the liver or kidney, which were the areas they were checking for damage. So it was also another successful result. Next, I want to talk about the methodology and results. So basically, the methodology was a literature review, and what I'm showing here is like a visual representation of the results. Basically, it starts with the lymphoma cell, and this is kind of just a process of how the entire nanoparticle, delivers the drug to the lymphoma cell. So it starts with the lymphoma cell, and there's antigens that are attached to it. And these, the next step of the process is that there's antibodies that attach to these antigens. And those are shown here. And what happens next is, so yeah, basically pre-targeting lymphoma cells use antibodies that are bound to these antigens. And what happens next is, this is basically the nanoparticle. And what occurs is when the nanoparticles are added to the antibody, it clicks. So this is what happens with the click chemistry. So the SPAC reaction occurs, and basically what happens is there's this triazyl ring, which is here with the nitrogen elements here that are combined, and through that are formed with the triazyl ring with the antibody, and then the drug is delivered inside of the lymphoma cell, which eventually leads to the death of the lymphoma cell. All right, so finally, I want to talk about the conclusion. So basically, I restated my question here. So can copper-free click chemistry using stain-promoted azide alkyne cycle addition be harnessed for nano-based drug delivery in lymphoma? And the results have been supporting this, and the results have supported this. And also what I found in the final, in all of, basically after doing all my research, I've also, in addition to supporting the use in this case for the CD20 antigen, there's also other antigens that can be used to harness with click chemistry, such as like the CD19 and BCMA. So it's not just limited to this to the CD20 antigen, which was used in this paper, which means that the findings are strongly supporting the use of click chemistry for targeted drug delivery in lymphoma. So finally, I want to talk about my experience with Gifted Gabber. Overall, I really loved the experience. I enjoyed it a lot. Learning about nanoparticles in the beginning to then discussing specific topics that I could potentially study, and then choosing a final topic and researching a lot about it. And I learned, specifically, I learned a lot about the creative writing process, as well as nanoparticles from Professor Virgil and Dr. Lilova. So I want to thank both of you very much, because I truly would not be able to do this without you, without all that I've learned from you. And I have also gained a lot of knowledge, about the the amazing potential of click chemistry, of copper-free click chemistry, as well as its use in the treatment for a non-Hodgkin's lymphoma. So that's my presentation, and thank you very much.
Awesome. Anish, you can stop sharing. I'll have Professor Lilova ask you a question. Professor Lilova? Yeah.
Very interesting topic. So what's your argument about the polymeric nanoparticles being part of the nanoformulations? Why did you choose them?
I chose polymeric because there are as well. I believe there's also a use of lipid nanoparticles. However, these are not very as effective, as well as I believe they're in addition to it's not as effective specifically in targeted delivery, because lipid nanoparticles, there's a potential for them to be accumulated in the liver, and so that's why that's a disadvantage of lipids. And so I wanted to study specifically polymeric nanoparticles, because they had more promising, based on research, they had more promising potential.
Thank you. Great.
Okay. Great job. A question from Professor Virgil.
Yes, congratulations, Anish. Job well done.
Thank you.
You're welcome. So this is in connection with Dr. Laloba's question. So what do you think is the importance of your study, and why do you think it's relevant to the scientific field?
Well, I think the importance of it is showing that this is a very promising, it's a very promising uh, potential treatment for lymphoma, for non-Hodgkin's lymphoma. And I think with further research, in fact, I saw that there's further research being done for other, not just non-Hodgkin's lymphoma, but lymphoma as well, and other types of cancers. And the contribution that this can give to the scientific field, and as well as to patients, is that it has the potential of being a less toxic and more effective way of treating patients with cancer, which is something that is a huge benefit, and a large benefit, because chemotherapy and current treatments for cancer are, even though they can save patients' lives, they are unfortunately toxic to healthy cells as well in patients. So that's why this can be something that can be a large benefit for patients as well, and to the scientific field. So yeah.
Excellent job, Anish.